really just sort of laid down the foundation of the connectome and how we got to this point. Today's session is going to focus on cerebral evolution and is going to delve deeper into the connectome evolution. This is an introduction on Dr. Porphyris. Dr. Christoph Porphyris completed his medical studies at the University of Cambridge with a double first-class pass and went on to complete his medical degree with distinction at Oxford University. He then did his neurosurgical training at the University of Witwatersrand and completed his neurosurgery fellowship through the College of Medicines. Dr. Porphyris has also completed the Charles Tier Minimally Invasive Fellowship under Professor Charles Tier at the Centre for Minimally Invasive Surgery and Neurosurgery in Australia. And during this time, he also undertook training in advanced brain mapping techniques under Prof. Mike Sohu, who gave us a lovely presentation last week. So I'm going to hand over now to Dr. Porphyris. He's going to take us through today's session, and we're looking forward to learning more about the connectome and cerebral evolution. Thank you so much.
Thank you, everyone. Thanks for joining us in this late evening timing. Okay, so today's topic, we're going to explore the connectome a lot further. So we're going to look at some of the basic science that underpins it. But before we do that, I will review some of the topics that we covered last week, and then go through a practical case, how we analyze the connectome, find problems with the connectome, and potentially identify targets that we use for TMS. After that, I'm going to go into brain architectonics, cerebral network development, and then the data science that underpins it. And we're going to finish off with the brain tracts, which is the first building block to understand how the brain is connected. Okay, so very briefly last week, we explained that we need 3 sequences to get a brain map using connectomic data. So the first sequence we said was a simple structural brain scan, a structural MRI. The second scan was diffusion imaging. So we used constrained spherical deconvolution, and then we mapped all the brain tracts in the brain. And that allows us to build a brain atlas. And then finally, what we discussed was functional MRI, and specifically the process of using resting state functional MRI to find which parts of the brain are connected at the same moment in time. We also covered the Glasser Atlas, and we said that this 2016 publication from data extracted from the Human Connectome Project was revolutionary because it was a brain atlas devised through machine learning, and it looked at aspects such as cortical thickness, myelination, functional MRI studies, and previous parcellation schemes. What we also said that every little segment in this brain is called a parcellation, and there's a total of 360 on each hemisphere. And this is drastically increased from Brodmann's 47 areas, of which only 23 remained. And we parcellated 97 new areas that we never knew about. So I'm just going to go in and talk about the networks. We talked about localizationism last week, and we explained how this has changed by neuroscientific thinking in the past few decades, that parts of the brain synchronize. They're wired together and they fire together during resting state fMRIs to bring about a common function. So what were the resting state functional MRI networks? Okay, well, there's 7 main networks from which we can build a lot of other networks. There's 2 main input and output networks, the sensory motor network and the visual network. There's a primal network, the limbic system. And then we talked about the cognitive networks, the default mode network, the central executive network, the salience, and the dorsal attention network. The sensory motor network, it's easy to understand. It's essentially the pre-central and post-central gyrus and the auditory cortex. On resting state function MRI, they all fire as one system. And as expected, it's responsible for sensation, motor function, and hearing. The visual network, we said, was the largest network with 52 parcellations, which is in keeping with our heavy reliance on vision as human beings. And its role is visual processing. And this is, as you would expect, in the visual areas, the occipital lobe. And we'll go into some more detail later on. The limbic network is part of the medial frontal lobes and the medial temporal lobes, and involves the Papez circuit together with these parts of the cortex, and it regulates emotion, memory, fear, motivation. Then we went on to discuss the cognitive networks. So some of the networks that we probably haven't heard of unless we've dealt with resting state functional MRI literature. And we talked about two competing networks: the default mode network, which is responsible for internal thinking and imagination, and the central executive network, which is responsible for external thinking. And what we said is these networks are in different areas. So the default mode network predominantly is in the medial frontal cortices, with the cingulate area in the cuneus region and the interparietal sulci, whereas a central executive network uses more of that dorsolateral prefrontal cortex and interparietal sulci. And what we said is they can never fire at the same time. So we're either using a central executive network for external thinking, or a default mode network for internal thinking when we're resting, contemplating how we fit into the world. And then we said there was a very large salience network with 49 parcellations that's predominantly in the anterior insular and the anterior cingulate areas, and that's responsible for switching between the two networks. Finally, we went on to look at the dorsal attention network, which involves bilateral frontal eye fields, interparietal sulci, and other association areas. And that's responsible for prioritizing our visual information. So we gave the example, if you're looking at your laptop now, your dorsal attentional network is telling you to focus on the laptop and nothing behind it. So what I'm going to do now is I'm going to exit the presentation, and then I just want to present a case. So I'm just going to go onto the Omniscient platform in order to give you guys an idea of how this works practically. So how does this translate to your practice? Okay, so currently, if you can see my screen, we're in the Omniscient platform. And what we've done is we've uploaded to the platform a structural MRI scan. So just looking at the brain structure. Then what we've done is we talked about the diffusion imaging, which loads all the brain tracts. So this is a young adult male who presented with a fairly long history of depression and tried multiple treatments, and was referred in conjunction with a psychiatrist for potential TMS. So that's the background of the case. So imaging wise, we saw the structural MRI, we saw the diffusion imaging. And what we do next is the case is uploaded to the cloud, and then the machine learning analyzes all of the tracts and builds this patient's personalized Glasser atlas. So what we've got here is 360 areas in both hemispheres, which is personalized to the patient because everyone's brain map is different. And what we can do with this, now that we have an outline of all the areas, is essentially understand his personal brain, and then we're going to analyze his resting state MRI in relation to this in a short while. The first thing I'm going to do is, since this is a case with the mood disorder, what we want to know is we want to know where the default mode network is. So the machine learning on the left here, we can pick out our tracts and networks. So we can toggle and find a network. Then on the panel, we've got different types of networks that we want to select in our patient. So what I'm selecting now is the default mode network. And what you can see here is a cingulate gyrus around that corpus callosum, the interparietal sulci, which make that network, and then those medial frontal lobes with the cingulate areas. And we can see the tracts, that cingulate bundle, which is connecting everything. The next network we want to look at is the central executive network for this patient. So just as a quick review, we've got dorsolateral prefrontal cortex, medial frontal lobes and cingulate areas, interparietal sulci, and some temporal areas. Now that the machine learning has told us where these areas are, we can do a connectomic analysis. So what we can do, we go to another panel, which is past the atlas, and we click on connectomic. And what we want to do is analyze the default mode network, and his central executive network. So we want to map the connectivity of these networks. And what we're looking here on the top left is called an anomaly detector. And I'm going to open it up so we can see it a bit better. What we've selected of all the 360 parcellations, because he's got a mood disorder, the best place to start is analyzing his default mode network and his central executive network. So we've got 27 parcellations in the default mode network and 24 in the central executive network. And we are comparing the connectivity of these parcellations to each other. As well as that, we're comparing them to a cohort of 300 normal young adult brains with no psychiatric or neurological disorders. And what we've got is 4 potential colors. So each parcellation is compared to another parcellation. So if we're looking at this one here, T1M is compared to 10D. Now, going back to the colors, if we have white, it means that the connectivity is normal when we compare it to our cohort of 300 adults. If it's black, it means there's too much variation between the normal cohort for us to make a conclusion. If it's red, it means that the connectivity between T1M and D32 is overactive. And if it's blue, it means that it's underactive. So what we can do is we've got essentially a brain QR code, and we can pick the areas that are over or under connected when we're comparing them to normal adults. And what this is doing in the background, it's taking the data from the resting state functional MRI from each parcellation and comparing those waveforms to the normal adult population. So the best targets to look for a potential TMS case is the columns with the most colors. So if we look here, we can see a red color here, and that's D32. And that's situated in the anterior cingular areas. So it's a deep area. So probably not a good target for TMS if we were considering it, because it's deep in the brain. And what we're trying to do with TMS is look for superficial targets. S32, again, it's a parcellation, which is below the GNU, so interhemispheric, very deep. PFM is a parietal target on the left, so it's potentially a good target, so I'm going to click it. And you'll see what we can do with the clicking later. T1M has 2 abnormal parcellations, so it's probably a better target. We've got 1 red one and 1 blue one, which we can click. Then the next one we've got is 8AV, that's situated in the posterior middle frontal gyrus, and that's a good target as well because it's superficial. The other 2 targets we have here is 10P, which is the frontal pole, and P47R, which is the dorsolateral prefrontal cortex. So potentially a target as well. Now, what I want to do is minimize here. And what our brain map does is it actually shows us where these areas are. So which areas have increased activity or decreased activity. So this one here is the 8AV. So if we look here, that's the 8AV, which was hyperactive in this patient. So potentially a good target to be suppressed. The PFM was a parietal target, so that's probably a good target as well. And that T1M was on the temporal lobe. So we've got 3 areas which we can now personalize to this patient and say, these 3 areas in your brain are not acting appropriately. And based on your symptoms, we do see this frequently with the mood disorders. So what we've done here is we use the personalized brain map to obtain personalized brain targets. And I'm just showing you this just so you can see it in action and how we can use the Connectome information to essentially administer personalized brain medicine. Okay, so I'm going to stop sharing and I'm going to go back to the PowerPoint. So the first topic we're going to cover today is brain architectonics. And it's basically looking back into evolution to try and understand the structure of the brain and how it's changed over time and how it relates to the brain as we currently see it. So if we look at the history of the evolution of the brain, we're talking about a process that's taken more than 600 million years. And the first evidence of any basic neurological structure could be found in the hydra, and it was in the form of nerve nets. So nerve cells, very disjointly connected to form very basic functions. And this wasn't really a brain. It was just the very beginning of what was to become the brain. The starfish then evolved to have a nerve ring with some radial nerves, but it wasn't until creatures such as the flatworms that we started getting cephalization, i.e. a concentration of brain cells around the head of the animal, where we started to see change in how the brain was going to start evolving. But the real beginning starts with the phylum Chordata about 600 million years ago. Now, to put this into context, to understand how long it's taken for our brain to evolve, the dinosaurs were around about 250 million years ago and became extinct 65 million years ago. So our brain's been evolving way before the dinosaurs appeared, and it continued to evolve significantly after they disappeared. Looking at it differently, Pangea, so 300 million years ago, there were no continents on the Earth. There was just a single continent called Pangea, and that was about 300 million years ago. So if you consider that it's in the last 300 million years, all the tectonic plates have caused continent migration, and the brain has only been halfway through its evolution since then, you can understand that the complexity that's evolutionary, you can understand how complicated our evolution has been to get to the point of where we are today. So if we go back to 550 million years ago and look at the first animals that showed some organized brain function, I'm going to start with the lamprey, about 550 million years ago. What we've got here is a coronal section of a brain. And what the lamprey basically had was something called the paleopallium. So that was the first brain, not a layered brain, disorganized brain cells. And what they started to do was surround an area of water. And this was seawater, more like sterile seawater. So this was the first type of brain. And its function was essentially olfactory. So it was to sense molecules in the water to potentially direct the animal towards a food source. So it was a smell brain. Now, the next step in the evolution was in the amphibians, and that took an extra 150 million years. So 400 million years ago, the paleopallium started evolving a bit further and started specializing. One of the specializations was the ganglionic eminence, which had a lateral process and a medial process. And this was the beginning of the basal ganglia, still on the surface, not deep in the brain. And the medial eminence gave rise to the globus pallidum as we see it today. And the lateral eminence gave rise to the caudate nucleus and the putamen. And the junction between the two is responsible for the septal nuclei. What else we can see is that some tracts, very rudimentary ones, were starting to project from this basal ganglia or ganglionic eminence type of structure. The next specialization from the paleopallium was the archipallium. And this is going to be the precursor of our limbic system. And from a single non-organized layer, it's the first cortex that started developing into 3 layers. And we can also see how all these structures are surrounding the ventricle. The next point in the evolution began with the reptiles, approximately 350 million years ago. And we started to see some change and some semblance of a brain that started to look a little bit more similar to our own. In addition to the archipallium and the paleopallium, the ganglionic eminence went deep into the brain and formed the subpallium, which is to become the basal ganglia. But then we also got a new 6-layer type of tissue called the neopallium. And that's going to give rise to the majority, more than 80% of our cerebral cortex. And if you note, it's very closely opposed to the subpallium, which is the precursor for the basal ganglia, which is roughly where the insular cortex is. So the first cortex to evolve was actually insular cortex. Again, we can see now that the archipallium, so that limbic system, is starting to migrate a little bit more medially, and the paleopallium is starting to migrate a little bit more medially. The other thing that we notice is that the tracts through the subpallium are becoming more pronounced, and they're actually connecting to the insula. So we're starting to get the start of the corona radiata, or all those tracts from the white matter that go through the internal capsule and split up the basal ganglia. The next step in evolution were the early mammals, approximately 150 million years ago. And what we actually see here is significant growth of the neopallium. So it's starting to shift everything and taking over most of the lateral surface of the brain, and also starting to exert some pressure on the paleopallium, so that rhinal cortex, the smell cortex. The ventricles becoming more opposed to the subpallium, or the future basal ganglia. More projection fibers are projecting through the basal ganglia. And we can actually start to see that hippocampal formation, or that folding of that three-layered cortex in the archipallium. Now, as we evolved further, the neopallium has taken over the entire lateral, superior, and inferior surface of the cortex, and starting to become gyrified, so having gyrification, as opposed to the leuencephalic brain of the early mammals. The archipallium has shifted completely medially, and this makes sense if we think about it in context of our cingulate gyrus, our isthmus, our parahippocampal gyrus, our hippocampus, which make up all that Papez circuit. And that's a three-layered cortex that derives from the archipallium. Our basal ganglia have gone deeper and maintain their relationship to the ventricle. We're getting stronger projection fibers, and our paleopallium is projecting much more medially. So it's going into that orbital frontal area where our rhinal cortex is situated. Fibers, of course, are projecting further. This part of the cortex, as we said, was derived from where the insula is. And interestingly, out of all the cortex, the insula is the only cortex that has a combination of 6-layered cortex, 3-layered cortex, and also older cortex, being the paleopallium. So it's actually got all types of cortex situated within the insula. So how does this compare to the adult brain? Well, what's very, very interesting is all the ancient parts of the brain as we've developed are sitting on the inside of the brain. So we've got the paleopallium, which is that orbitofrontal gyrus, which is essentially 550 million years old. We've got the subpallium, which is approximately 400 million years old, sitting where the basal ganglia is. We've got the archipallium, where that cingulate gyrus is situated, which is approximately 350 to 400 million years. Whereas the neopallium, which has evolved on the surface of the brain, it's much more recent. It's less than 50 million years old, or approximately about 5 million years old. So when we're actually looking at the human brain, when you look more closely, there's a lot more evidence of architectonic design of the brain. For instance, the cortex around the cingulum is much more older than the surface cortex. And when you look on T2 scans, very often you'll find that the intensity is brighter on the cingulate cortex as opposed to the surface. And that's got to do with its evolutionary origin. And in fact, there's some very interesting work which shows if you draw a circle around all these structures, you're actually looking at the ancient brain versus the new brain on the outside. Understanding this just gives you a much deeper of an understanding of how the brain works. And for instance, now we know why, for instance, our limbic system is situated on the inside of the brain medially, because it evolved from an ancient system where it got shifted by the evolution of the younger brain on the surface. All right, the next topic we're going to cover. So there's a few disparate topics, but you need to cover them in order to understand how everything ties together and how we're bringing a lot of different aspects of science together to build on our connectome knowledge. So next I'm going to talk about graph theory, because it's really revolutionized the way we can analyze brains. And essentially, it's given justice to the complexity of the brain and given us a scientific method to study it. If we look at what graph theory is, it's essentially a study of graphs that model pairwise connections between different items. And its origins start with Leonhard Euler in the 1700s. He was a Swiss mathematician, And there was a problem in Königsberg, which was Prussia at the time, but modern day Russia and Kaliningrad. And the problem in that area was that there was 4 land masses, which were separated by water. So one landmass, another landmass, another landmass, another one. And there were 7 bridges in between. So the problem was they wanted to know, is it possible to go through all of the land masses with only passing each of the bridges only once? And there was a lot of debate and a lot of arguments, and no one was really sure. So what Euler did is he came up with graph theory. So instead of looking at all the complexity of the landmass, the water, the trees on the banks, the bridges, he basically created nodes, so little circles which represented the landmasses. And then what he did is he modeled the connections, which were the bridges. So these are called edges. So nodes are connected by edges. And then this led to a graph. So we can perfectly look at this problem, simplify it, and then look at it as a graph. In essence, the answer to the question was it wasn't possible to walk through all the land masses passing the bridges only once, because in order for it to work, each of these nodes have a different number of edges. So in order for that to work, you had to have 2 nodes or less with odd numbers. And it works out that all 4 nodes have odd numbers. But what it gave rise to is it gave rise to a very sophisticated way of looking at complicated problems. So how do we apply this to the brain? Why is it very, very attractive to use graph theory for the brain? Well, one, because it's very complex. So instead of us trying to think of neurons and axons and functional MRI and all these different elements, what we can do is we can decide how to break up the brain and treat that as nodes, and then study the connections between the nodes as edges. And what's classically done in connectomic literature is there's 2 different ways to break down the brain in nodes and edges. There's an anatomical method and a physiological method. So with the anatomical method, you can break up the brain into lobes or sublobes, or what we're doing with a Glasser atlas, where we're using that diffusion imaging to break down the brain into 360 areas. And then what we can do is you could study their physical connections, such as axons, and then through the tractography, and compute the edges and then study the relationships with graph theory. Conversely, you can look at the brain through fMRI. So you can pick different points and look at the time series data from the fMRI studies, and then look which parts of the brain are functionally connected to other parts. And then again, you can connect those relationships with edges and study that. So what are graph theory basics? So what I've told you so far is, graph theory is an important mathematical method of breaking down the brain in parts and studying the relationships. And it's allowing us to study the brain with maths, which is much more sophisticated. So some of the graph metrics that we can look at to study the different networks is something called degree. So the degree of the node in this basic network and in any network is how many times is a node connected to another node through an edge. So if we were to study, for instance, A, the degree of this node would be 2. If we were to study B, the degree of that node would be 3, 5, 6, 7. So the degree tells us how many connections a node gets. From that, we can derive a metric called network strength. So what network strength does, it calculates the degree of all the nodes in the network and tells us, on average, how many degrees does each node have. So if the nodes have more connections, it's a stronger network. If they have less, it's a weaker network. We can look at something called the clustering coefficient. So what that does is, if you've got a region with different nodes, it tells you on average, what's the chance that this node here is connected to its neighbor. So you can get a coefficient that will tell you, based on the connectivity of that network, how likely is this node going to be connected to that one or that one. Another thing that's very important that we can look at is called the shortest path. So in a network, we want to know how efficient it is. So if you wanted to get all the way from A to D, in how many connections or how many jumps can you get there? Because if we can go from A to B, from B to C, from C to D, it's the shortest path to that area, and it looks fairly efficient. But if we didn't have this connection, then we'd have to go A to B to this green node, then to C, then to D. So it would have a longer, shortest path. And it's very important because we can calculate a path length for the entire network. So what we can do is look at all the connections of the nodes and the edges and mathematically calculate what's the average shortest path for the entire network. And that has huge implications for how good or how bad a network is. And it's very important in the way we study a lot of diseases in the brain. Centrality looks at how important is a node. So what it does is it'll take a node and it'll analyze its neighbors, how it's connected via edges, and then it'll also analyze how the neighbors are connected to other nodes. And there's lots of different ways to calculate centrality, but essentially a node with a higher centrality is more important than a node with lower centrality. And it makes sense. So if you look at this node here, which has a lot of connections, if you take it out, you basically wipe out this part of the network's connection to the rest of the network. Whereas if you took this one out, then there wouldn't be much of a problem. You only take up one edge. When we're looking at more global properties for a network, we can look at something called the module, and that just breaks down the network into different parts that are working independently. So you can see that around the red circles. We can talk about a hub. So a hub is a node that connects to other parts of the brain. So again, going back to this example, we've got a node which is acting like a hub, and it's able to connect this node to that one and then communicate with this module. Finally, another definition that we've got is a rich club. So the saying is the rich gets richer, and this actually applies to social networks as well, the internet and lots of other applications. But basically what we have here is a collection of hubs that are connected to each other. And because they're all connected to each other, they're making a rich club. Now, I know this might sound a bit arbitrary, but what we've got here is the building blocks to study the brain through graph theory. So if we want to understand different parts of the brain, we can use this and apply maths to neuroanatomical and neuropathological problems. For instance, if we study path length, so remember we said path length is on average for the entire network, how short is the communication between the nodes? If we study path length and apply it to IQ, so someone's IQ, it's been clearly shown in studies that the shorter your path length is, the higher your IQ is. So what it means is that people with higher IQs probably have a more efficient network, so able to transmit data between their different networks quicker because they've got shorter path lengths. So they're probably making less connections or less synapses to get there. We can look at centrality. So centrality tells us what are the major nodes in the brain, which ones are the most connected. And this is a very interesting study by Mike Shagru, actually. What he actually did is he broke down the brain into the Glasser parcellations as we've done. And then he wanted to know which of the nodes, so which one of those parcellations, which each one was a node, is the most connected essentially. So which one is the most centrally connected? And what he used a metric called PageRank centrality, which is the same algorithm that Google initially used to grade web pages on their search engines. And what we found is that by and large, the majority of the parcellations with high centrality are eloquent parts of the brain. So if you look here in this subject, V1, 2, and 3, it's all primary visual areas, primary motor cortex, premotor areas, of Wernicke's areas. So what we can see is we can define brain eloquence by centrality, which is a graph metric. Finally, we can also look at the different networks to look at the clustering coefficient. So how connected is a part of the brain, is a subnetwork of the brain to each other. And an interesting paper looked at all the brain networks that I discussed about earlier, such as the default mode, salience network, and they looked at it in terms of aging. And what happens when we age is that our networks become less segregated. So instead of talking more to each other, they talk less and start talking to other parts of the brain. So it's thought that part of our cognitive decline with aging is because that clustering coefficient is decreasing in size. The next thing I'm going to talk about is a small world network. And this was a concept published in 1998 by Watson Strogatz. It's actually been cited more than 46,000 times, and it's probably one of the most important papers in science. What they did is they were studying networks, and they looked at a regular lattice matrix. So what a lattice is basically nodes next to each other, which are directly connected to its neighbor. It's ordered and organized. What they then did is they used a computer model to randomly delete some of these edges, so the lines connecting the different nodes, and then connect them to further targets. And what they noticed is that a regular lattice has a very high clustering coefficient, which means it talks to its neighbors very well, but it's got a very, it doesn't have a short path length. So what that means is it's not good at talking to someone on the opposite side of the spectrum. So if you're the brain and you're the frontal lobe and you want to talk to the occipital lobe, you're going to have to jump here and jump there and jump there and jump there. So you've got a very long path length. And this is not an efficient network. But if you delete some of those nodes and now change those connections and do some cross-chord connections, what you're doing is, if this part of the brain, which is the frontal lobe, has to talk to the occipital lobe, we've got a direct connection. So what we're doing is we're drastically reducing that average path length and making it short. And because we're only making some minor deletions, we still have very high clustering coefficients. So we still have a very efficient network that's able to subspecialize in smaller networks. And this is called the small world network. By contrast, if you randomly delete all the edges and connect them randomly, you can get a short mean path length, but you lose your clustering coefficient, so the ability to function in local networks. And this underpins how the brain is connected. Because if you think about it, we've got lots of specialized areas with short axons between them. And that's efficient communication, because you're not going to spend a lot of energy communicating between two neurons that are close to each other. However, in order to integrate as an entire network and make us who we are, we do need some communication which is far-reaching, such as the white matter. But that can only be done if we've got long tracts, which we're going to study later, which are efficient at changing information from one side to the other. And what's interesting with this, if you study the internet or a social network or many other complex systems, they're all governed by this principle. So it's almost that all these complex systems are using unified mathematical problems, principles to solve a problem of communication. I'm going to go on to talk finally about complex systems. So there's an entire field called complex systems. And what it does is it studies large networks of interconnected and interdependent areas and their adaptive parts. And they've got some special properties, and the brain is a complex system and it shares these properties as well. So one of the properties is that it demonstrates is something called emergence. So what emergence means is that you can break down a complex system, i.e. the brain, into axons and lobes and synapses. But at some point, even though you can break it down to all its parts and understand how it works, you can't explain at a macro level how it comes about to more complicated functions. So we know how the brain works, but we can't really explain how it's going to make someone compose a beautiful piece of music, or how it's going to make someone listen to that music and recognize it and appreciate it. And that's called emergence, where we're at a level of understanding where we can't really explain the more complicated functions that come out of a complex system. The next property is that it shows something called power law distribution. So a lot of statistics that we look at are governed by a Gaussian normal distribution. So for instance, if we look at height, an average height would be 175 or 170. And then there's two arms where we might get people on the extremes of that normal distribution, where someone's 2 meters tall, 2 meters 10, you might get someone 2 meters 20. And then you might get really short people that are 1 meter. but you're never going to get someone who's 7 meters tall. So in the history of mankind, it's very unlikely that anyone's been 7 meters tall. And that's limited by this normal type of distribution. With a power law distribution, what we've got is that you can have a lot of people who are, for instance, 1.70 meters, but the way the distribution is, it's not impossible for someone to be 10 meters tall. And this applies to the brain and brain connections. So the vast majority of the connections are situated in a local network where you've got short synapses, but it's not impossible to have a corticospinal tract whose axon is more than 1 meter long. So complex systems tend to show this power law distribution. Finally, complex systems show robustness. And what we mean by that is if you start attacking the network, so if we look at a brain injury, if someone gets a small injury in a small frontal lobe area, we're probably going to delete some of those nodes because they're not working, but the network's able to carry on and essentially still function. And that's called robustness. However, because as we explained before, some nodes have higher centrality or they're hubs and they connect to other important hubs. If you did a targeted attack on that network where you're just hitting the central, the nodes that have high centrality, i.e. the eloquent areas of the brain, you get much more devastating injury where the network's not able to compensate. So what I've covered is just some different topics. I mean, you don't need to know all of this to essentially look at a patient's connectome and understand how it applies in the clinical setting and translational medicine. But I'm just trying to introduce everyone that there's a lot of science that's gone behind this. In the last 10 years or 15 years, neuroscience has gone leaps and bounds, and it's just understanding the brain in a much more sophisticated manner that actually gives it justice to its complexity. Okay, I'm just going to briefly talk about brain network development. So if we look at the human brain in the first year of life, the volume of our brain doubles. And it's only until about 3 we get to about 90% of our adult volume. So one interesting question is, how are all these networks that I talked about before, those 7 networks, how are they developing? What's their time frame that they kick in? And can it explain a child's development, for instance? So what's very interesting, from birth till about 2 years, we've got development of the somatosensory and auditory network and the visual network. And they're probably present from birth. So when you actually look at the functional MRIs and you look at the connectivity, the somatosensory cortex and the visual cortex almost shows connectivity that mirrors adult connectivity. And that makes sense because when you're born, you do need to feel things, you do need to try and move. So that network needs to be advanced fairly quickly. The same with vision. You're starting to learn how you see things. So it's not a network that can develop later. However, when we look at the cognitive networks, the default mode network, the central executive network, the salience, the dorsal attention network, they're not connected as a network. So you can actually, on a functional MRI, look at their different parts. So if you look at a default mode network, we said that it's got a posterior cingulate area, an anterior cingulate area, and an interparietal area. All those 3 areas are functioning independently. So they're not on a resting state functional MRI integrating with each other. And it's only until later in life and onwards that those networks come online. And that's what we think is leading to our maturity as we mature through adolescence. The default mode network probably comes online at about 2 years of age. And it's very interesting because that's about the same time that children start to recognize their own reflection in a mirror. And remember that network's responsible for internal thinking. So putting yourself, putting the idea of yourself into your environment. So understanding that you are part of your environment and you're a part of that. And that's when kids recognize their reflection, which reflects that. Okay, so if we just look here, we're looking at cortical thickness. So on the left, we've got brains from infants that have just been born at 1 year and at 2 years. And what we can see here is cortical thickness, which jumps from 1 millimeter to 4.5 millimeter. And we can see that when we're born, there's very few areas that are very thick, especially none that are going to the 4 millimeter level. But we can see in those first 2 years that the medial frontal lobes, which also have a lot of that default mode network in there, increase in thickness drastically. Now, these are graphs of brain regions. So this is from a neonate, that's from a 1-year-old, and that's from a 2-year-old. And what we're doing is we're modeling the different lobes. So that's a frontal, that's a temporal, that's an occipital lobe as graphs. So we're looking at the nodes within that lobe and how they're connected to each other. And what you will note in a neonate is that the frontal lobe, it's all blue and it just talks to itself. The temporal lobe is all yellow and essentially just talks to itself. So there's not much communication functional wise between the different lobes. But as we get to 2 years of age and we start looking at the fMRI data, so looking through the fMRI, which different lobes are talking to each other at the same time, we start to see that structurally the frontal lobe is here, but it's also able to communicate to different lobes. So structurally the temporal lobe is here, but it's able to communicate with different lobes. And that's starting to show us that the networks are coming online. So all these networks that we're describing, instead of being isolated to one anatomical place as an infant, then they start to slowly connect with other parts of the brain. And that's got to do with how we're myelinating and a few other processes. Again, this sort of touches on the same topic. So we've got nodes from functional MRI data in a 1 to 2-year-old and in adults. And what we said, we'll just focus on the default mode network. We said that it's posterior cingulate area, anterior cingulate area, and the interparietal areas. And what we've got is the anterior cingulate areas are just talking to themselves and not communicating with the posterior cingulate areas, which are a different color, which are not talking to the parietal areas, which are a different color. But when we look at the adult, it's the default mode network's all red. So when you're looking at functional MRI data, what it's telling us is that the anterior cingulate is talking to the posterior cingulate to the interparietal sulci. So it's all working as one network. Finally, we've got a graph that's showing us the participation coefficient. And what that's telling us is how effectively are the different nodes within a network talking to each other. So if we look at all the 27 parcellations in the default mode network, when we look at a graph theory metric participation coefficient, how efficiently are they talking to each other? And we're doing this for different networks. Now, what's very interesting, when we jump to adolescence, so from about 12 to early adulthood, the network that really comes online is the salience network, which we said is a switch network between the default mode and the central executive network. So at 12, the participation coefficient, i.e. its ability to talk to itself as a network, is low, and that it increases drastically, especially when you compare it to the other networks. So it definitely has an important role in terms of us maturing into adults. Okay, what I'm going to talk about next is the white matter tracts of the brain. And they're important because they're connecting all those 360 parcellations that we talked about to each other. If we look at small world networks, it's the way the brain makes long jumps to different parts of the brain and is able to make all the networks efficiently communicate with each other. And what we're going to do is in the next few weeks, we're going to break down all the lobes and the parcellations and talk about the different parcellations and what they do. But in trying to understand the parcellations, you've got to understand what they're connected to. It's a bit of an encyclopedic task, so I don't expect everyone to remember everything and know it by heart. And you don't need it to process your patients and understand how connectomics applies to a clinical problem, but it's just trying to show the science behind the whole process. Now, part of understanding how these tracts work is also understanding which parts of the brain are connected. And this is what's changed with the study of connectomics. So before we knew about a few of these tracts, not all of them, but we never really knew what they connected. Whereas now with connectomic analysis, we know where they're connected. So the first tract is the most important one, which I'm going to look into, and that's called the superior longitudinal fasciculus. And that's a tract which is the largest tract of all the tracts. And what it does is it rotates around that Sylvian fissure, and it's important in connecting the frontal lobe, the parietal lobe, and the temporal lobe. And if you look at it, it's almost like a Y on its side. It's got a frontal ramus, a parietal ramus, and a temporal ramus. And in fact, it's actually split into 3 parts. So SFL1, SFL2, SFL3, and the arcuate fasciculus, which connects Broca's to Wernicke's, is part of that SLF3. So if we look at SLF1, what it does is it connects a superior parietal lobule to the premotor areas of the brain. The SLF2 connects the inferior frontal lobule to the middle frontal gyrus areas. And then what SLF3 and the arcuate do is they traverse through the frontal and parietal opercula, connecting that posterior temporal parietal area to the inferior frontal gyrus. And this is just a representation of the parcellations that are involved in this tract. And you can see it's 64 parcellations, and it's a huge tract, which pretty much connects all of our hemispheres to each other and leads to efficient information transition. What does it do? I personally like to think about it as a highway. So a lot of the literature tries to focus on language, but it does a lot more than that because it's connecting 64 parcellations. But if we're going to give predominant functions, on the left side, it's responsible for phonemic language. So the way we actually are able to say words. So problems with it can lead to phonemic paraphrasias. And later we'll discuss about the language system. There's a dorsal and a ventral language system and how the SLF fits in. On the right, it's probably important in the attention network, the praxis network, and neglect. And these can all become problems when there's injury to the SLF. If you really want to go into detail, this is from one of Mike Chagrew's publications on the superior longitudinal fasciculus. Essentially what it's showing is you can trace all the tracts and figure out which parcellations they're connected to. And it leads to a ridiculously complicated scheme, but the information in it is huge because it allows us to study neurological problems in a way that we were never able to do before. Okay, the next tract we'll talk about is the medial longitudinal fasciculus. And what that does is it connects the posterior medial occipital lobe to the superior frontal gyrus and part of the superior temporal gyrus and the temporal pole. And this one has 28 parcellations, and then we can see here the occipital areas and the temporal areas it connects. And it's actually a pretty small tract, which is discovered in the 80s. Not much is known about it, but there's some thought on some studies that it leads to converting a sound to actual meaning in our brain, and also potentially responsible for us learning new words. The next tract we'll talk about is the inferior longitudinal fasciculus. So we've gone through a superior longitudinal fasciculus, a middle, so there's also an inferior. And what that connects is the parts of the occipital lobe to the inferior and medial parts of the temporal lobe. And it's a larger tract than the middle longitudinal fasciculus of 33 parcellations. And we can see here which parcellations are involved in the lobe. But this one's very interesting. It's actually very important in object recognition, face recognition. So it plays part in that ventral system of the occipital lobe when we're looking at visual. So it's the ventral visual processing system, or the ventral stream. It's also got involvement in memory and semantic language. Now, what's really interesting, and we'll see later on, there's parcellations in the brain which are connected by this inferior longitudinal fasciculus, which connects the occipital lobe to that medial frontal lobe. And in the fusiform gyrus, there's areas that have been studied through fMRI in people who were born blind, so they've never seen a face in their life. But when you describe faces to these people, it's those areas of the brain that you would expect in someone that can see to light up when they see a face. When you just talk about a face, it lights up there. And what it shows is that the architecture of the brain is genetically determined. So the place that's supposed to see a face will light up in someone that hasn't seen it before. And that's just very interesting. The IFOF, or the inferior frontooccipital fasciculus, is also a very important tract. And what it connects is the inferior frontal lobe. It passes through the extreme capsule and then goes into the medial frontal lobe. And its role is in semantic language. reading, writing, and some goal-orientated behavior. Now, importantly, when we think of language, we've got a ventral stream and a dorsal stream of language. What the ventral stream does through the IFOF is to give meaning to language, and that's the semantic pathway. And that's what's been explored by awake craniotomies very frequently. The dorsal pathway of language is the how. So how do we put words together? And that's the information from that part of the language system is communicated by the superior longitudinal fasciculus that we described earlier. So there's two different streams of language. It's a little bit more complicated than that with some areas in the temporal pole, but as a basic model, it sort of describes the function of language. Now, the cingulate bundle is a very important bundle. If you'll remember back to architectonics, we said that's coming from the archipallium, so three-layered cortex. So it's situated in that cingulate gyrus, going through that isthmus, parahippocampal gyrus, going into the hippocampus, and then connecting through the Papez circuit, mammillary bodies, anterior thalamic nuclei. And then again, we see the parcellation that it involves. So it's all those areas that I mentioned earlier. And it's responsible, as expected from the limbic system point of view, for emotional processing, memory. But very importantly, if we think of the brain as a default mode network, which Papez didn't know when he was describing that, we've got an anterior cingulate area and a posterior cingulate area. And it's a cingulate bundle that's actually connecting the two. That's what we saw earlier when we went through the omniscient platform. So it's also got a role for the default mode network functions of internal thinking and imagination. The uncinate fasciculus is interesting. So what it does is it connects the medial and inferior frontal lobes to the temporal pole. Now, you remember earlier from the architectonics that the paleocortex was the smell brain, the rhinal cortex, which is also situated in this area. But the uncinate fasciculus is probably a very old fasciculus. And because of that, over evolution, it's acquired a lot of functions, not just the ancient functions. It doesn't even have many parcellations, only 9 parcellations, but it's got a role in semantic language on the left side and memory. On the right side, studies have shown that it's involved in empathy, or our ability to have self-awareness and how we fit in. And on both sides, there's studies that show it's responsible for emotional regulation or impulsivity. Now, what's interesting, even though it's involved in these language tasks, you can do a temporal lobectomy, and I do it fairly frequently for temporal glioblastomas, and you won't get an obvious language problem. We've also got the frontal ascendant tract. So this was only discovered in 2008. We didn't know about that previously. And very interesting, what this does is it connects the supplementary motor area to Broca's area, so the inferior frontal gyrus. And what it does is it allows us to initiate speech. So we make a speech plan in the SMA, the information goes through the FAT, or the frontal ascendant tract, to Broca's area. So injury to this can lead to stuttering or verbal fluency problems. The vertical occipital fasciculus, or VOF, is interesting in that it's an internal occipital lobe connection. So it connects the inferior lateral part of the occipital lobe to the superior part of the occipital lobe. So it's not near the primary visual areas. And its role is thought to be important in integrating the visual streams, or the stream informations for vision. So remember, with vision, we've got a dorsal stream and a ventral stream. So the ventral stream tells us what something is. I recognize the face, recognize a tool. And the dorsal stream tells us where something is in space. But even though this information is separating, we do need a way where we're integrating this information to get an experience. And it's thought that the VOF, which basically communicates the two streams, is that integration. Finally, for completion, I won't talk much about it because I'm sure everyone knows about it. We've got a corticospinal tract, and that's not technically a connection within the brain. It's going through our motor areas into our spinal cord, and there's a few other functions. Just to demonstrate the power of knowing these tracts, and it's not just airy-fairy sort of anatomical intricacies. So very recently, I've had a young guy who's presented to us with with the first onset seizure as an adult. And what he's got is on his MRI, he's got a hyperintense lesion in that posterior frontal lobe. And this is very suspicious for a low-grade glioma. And the thing about low-grade gliomas is if you do an aggressive resection, you can really extend many years of life for patients because they have the tendency to become a glioblastoma. So if you're very aggressive with your resections, instead of a patient dying a few years after diagnosis, they can potentially go many decades. So we did the tracts, and all we see here is a pretty picture. So I've talked to you about these tracts. The red is the tumor. Now we're seeing the corticospinal tract, the SLF, the IFOF, and it's nice. It's a pretty picture. But when we look in more detail, what we can see here is the tumor's right next to that FAT, that frontal ascending tract, that supplementary motor area connecting to the inferior frontal gyrus. So if you're going to take this out and you injure that, he's going to have serious speech problems. Also fairly close to the corticospinal tract. So this is a potential case that will warrant potentially an awake craniotomy to try and mitigate that. But it's just an illustrative example of some of the practical applications of the tracts. So there's a lot of information there. As I said, it's just to try and demonstrate some of the science behind all this connectomic knowledge. And that's the end of this talk. And next week, we're going to be going into the frontal lobe and looking at all the parcellations. Thank you for your time, everyone.
Thank you so much, Christos, for that. So we'll open up the floor now to any questions. You can either unmute or you can type it in the chat. I think there was a question typed in the chat earlier by Fazila asking for you to comment on the connections in autism.
So look, autism, I haven't studied specifically. I know some of the basic studies are showing that they're not as well integrated as what they are in a normal adult brain. We will have Mike Shagru on the last talk again, and he's doing a lot of work with a few autism groups looking at those connections specifically. Again, there's a lot of work and you can do multiple PhDs on it, but I can't give you any more detail than that, unfortunately.
Okay, thank you. Are there any other questions? Okay, it looks like there's no more questions at this time. Thank you so much for today's session. It was, again, very, very incredible. And I think just gaining and understanding all of this has just been really, really eye-opening. Just as a reminder to everyone that there will be another session next week at the same time. You can use the same link to join. And as Dr. Price has said, next session is going to focus on the frontal lobe and the parcellations within the frontal lobe. So looking forward to delving deeper into that. But otherwise, thank you so much for tonight. Again, very, very insightful. And looking forward to the next session.
Pitsy, it's Mandy. Can you hear me?
Yes, go ahead.
Hi, Mandy.
Just at the top, I put the link for last week's talk, if anyone wants to download it. Otherwise, you can go to YouTube and type in Netcare YouTube and you'll find the talk as well. And we hope to post this one tomorrow or the next day as well. Thanks very much, Chris.
Thanks, everyone. Appreciate it.
Awesome. Thanks, man. Thanks, everyone. Have a good evening.
Thank you.